Hello, biologists. Today we're going to talk about transcription and translation, which is what we use to make a protein. So we're going to create a protein. We call that protein synthesis. The first step is to identify a gene. A gene is the piece of DNA that you use to make a protein. So you have a gene for hair and skin and liver enzymes and your immune system. All of our proteins are created with these genes here. Once you find the gene, you're going to have two sides to it because we have a DNA double helix. One side is going to be the template strand, and one side is going to be the coding strand. The template strand is the strand that is used to create an mRNA. Remember, your DNA is 6 feet long and it's inside of your nucleus. It's too big to leave. And so since it can't leave, we need to make a copy of the information, and we make a copy of the information in the form of mRNA. So transcription, turning the information in DNA into mRNA. That's our first step. This occurs in the nucleus, and we do this by transcribing genes. The transcription of mRNA is going to use complementary base pairing so that your template strand of DNA will create a complementary pair of the information it has there. Remember, since we're making RNA, we're going to use U's instead of T's. We're going to have uracil. Then when you have a piece of mRNA, it's going to be modified a little bit as it leaves the nucleus. And then it will go over to a ribosome, the organelle that creates proteins. And the organelle ribosome will read the piece of mRNA and turn it into a protein. That is translation. How that works, you have a 3-nucleotide sequence in your mRNA that's called a codon. Each 3-nucleotide sequence is going to relate to one amino acid. And so the sequence of nucleotides in my mRNA determines the sequence of nucleotides I put into my protein. This is the primary structure of the protein. The charges on those amino acids will cause your protein to either do an alpha helices or a beta pleated sheet, and those will create the secondary level of organization in your protein. And then your 3D structure of a single protein, how those charges and how those bends and spirals make it glom together into a 3D, that's going to be your tertiary structure of your protein. We would have a quaternary structure of a protein if more than one protein component adhere together, but not all proteins have a quaternary structure. Let's look at transcription in a little bit more detail. Transcription is the process of making mRNA. We're going to use the information in DNA to produce it, but note that DNA does not turn into mRNA. DNA stays where it is. It is not destroyed in this process. We use the information to create complementary base pairing to the template strand, creating mRNA. So this is created, but DNA is not destroyed. There are 3 steps to this process: initiation, elongation, and termination. Let's look at initiation. That's what we see on the right here. The first thing that we need to start transcription is we need a promoter. A promoter is a piece of DNA in which transcription factors, which are a bunch of enzymes and proteins, will bind. In eukaryotes, there's often a repeat of T's and A's. So it'll be TA, 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 TA. And so we refer to it as the ta-ta box. The ta-ta box will help you identify where the promoter is. Transcription factors will bind to that ta-ta box, to the promoter, and then the enzyme that will separate the 2 strands of DNA and start to piece together RNA nucleotides to create mRNA will bind right on top of those transcription factors. So think of it as like the landing site for the enzyme that creates mRNA, which is RNA polymerase II. Now, as DNA polymerase III, when we are doing DNA replication, both transcription, we're creating RNA, so it's going to be RNA polymerase, and it's RNA polymerase II because that's the second one that was named. Here we have our initiation begun. Let's talk about elongation. In elongation, your RNA polymerase II, which is this orange structure, has already bound to the transcription factors at the ta-ta box using the promoter to help it figure out where to go. It's going to now unravel the DNA helix, and as it moves through RNA polymerase, it's going to access the template strand. The template strand is going to be used to make a complementary base pair to those nucleotides. This RNA polymerase II will run down the DNA until it gets to the termination site, and then it'll let go of the DNA. So picture this enzyme moving to the right as we go. That's initiation. In elongation, RNA polymerase II is taking nucleotides, the ones that you would use for RNA, so it's uracil, not thymine, and it is binding them together with those phosphodiester bonds, creating a single strand of messenger RNA. This single strand of messenger RNA is complementary base paired with the template strand. Now, the other side is called the coding strand, and the reason it's called the coding strand is because it's also complementary base paired to the template strand because it's the other half of DNA. So if you were to compare the sequence of mRNA to the coding strand of DNA, it would look the same, except mRNA would have U's and the coding strand would have T's. So the template strand is what we use to make it, but the coding strand and the mRNA look similar, which is why that one's called the coding strand, because it kind of has the code. This mRNA will continue to be created until we hit the termination site of DNA. When we do, DNA or RNA polymerase II will disassociate with the DNA, and you can now move on to the next step, which is translation. The mRNA has to be modified before we can begin translation. To modify RNA, we make it mature. So the first thing you do is you remove the introns. Introns are pieces of genetic information that are not actually used to make an amino acid. So they are there in the gene, but they're not used for protein production. Their evolutionary history and why they exist in the first place is something that is still being studied, but what we do know is that we have to get rid of them. The way we get rid of them is with an enzyme called the spliceosome. And what the spliceosome does is it takes your mRNA and it squeezes the ends of two exons, which are the pieces that we do want, close together, and then it cuts out the intron so that we are left with only exons of mRNA. Here's another visual of this. This is my immature mRNA. It contains introns and exons. I want to get rid of the introns. So the spliceosome will form on opposite sides where two exons are joined, and there's an intron in the middle. It'll come together and it'll kind of squeeze out the intron, and then it will cut it and reconnect the mRNA. So there's going to be a phosphodiester bond added, and you are left with just exons, and the intron itself is removed and disassembled. The word laureate means lasso. Someone thought this looked like a lasso here. Another thing that we're going to do to our immature mRNA to make it mature is we're going to add a poly A tail. You add this to the 3' end of the mRNA, and it's just a sequence of A. On the 5' end, you add a 5' cap. Both those things prevent degradation as it moves from the nucleus to wherever the ribosome is located. Your ribosome is going to have two components. It has a large subunit and a small subunit. I think of it as like an unequal hamburger bun. So it's got a puffier top and a flatter bottom. In your large subunit, you're going to have 3 sites. You're going to have the E, a P, and an A. Think like Environmental Protection Agency. The E site is almost always empty, E for empty. The P site is where we're going to have the growing polypeptide chain. So P for polypeptide. And the A site is where we add new amino acids to the growing polypeptide. Those amino acids are attached to tRNA. tRNA stands for transfer RNA. At the top of your transfer RNA, you have an active site that binds to passing amino acids. And then this transfer RNA binds to the A site in the ribosome. Your ribosome will kind of shift. And as it shifts, the polypeptide that's attached to the P site will dissociate and it'll reassociate on the new amino acid in the A site. So as it shifts, the transfer RNA that was in the P site is now empty, and it gets scooted into the E site, and it will be ejected from the E site. What was in the A site is now in the P site, and the A site is empty again, so you can add another transfer RNA with another amino acid. This will continue as it shifts down the mRNA, and it will read one single codon at a time. At the base of tRNA, there is a series of nucleotides attached. This is referred to as the anticodon region. The anticodon region, which is right here, will attach to the codon in the mRNA. So those two things adhere together, and that is how your tRNA knows which codon it's binding to, because it is a complementary base pair with the anticodon region on the tRNA. Here is the A site, where the new amino acid is being added. This amino acid is shown right here, attached to the transfer RNA. As it adds itself, this ribosome will scoot forward. The polypeptide chain that's being shown here, each of these is an amino acid, is going to detach from this tRNA, and it will attach on top of this one. And then the now empty tRNA here will be ejected from the E site. So I have my E site, my P site, and my A site right here. Let's look at another visual of that. Oops. Here I have my mRNA. My mRNA is going to start with the same codon each time. It's called the start codon. It is AUG. AUG always pairs with the anticodon region of a tRNA that is holding a methionine. So every protein starts with MET. So to begin initiation, the first step of translation, you're going to have your large ribosomal subunit and your small ribosomal subunit sit right on top of the start codon, and you will bind the first tRNA right here with MET. Then your ribosome is going to shimmy down, and as it does, a new tRNA will join into the A site. This is the E site. And then here is the P site. A new amino acid carrying tRNA will attach to the A site. We will know it's the right one because its anticodon region will bind to the mRNA at the codon that's in there. And then your ribosome will shift over, and this polypeptide here will attach to the to the amino acid that is on top of the newest tRNA. This amino acid will be moved, which means that this tRNA is now empty. It will get pushed into the E site, and because it's empty, it will just leave the E site. That means that what was in my A is now in the P, and the newest amino acid is at the bottom of the growing polypeptide chain in the P site. Here's a visual of it. I have my growing polypeptide chain right here. I'm going to add one more. To add one more, I'm going to take this polypeptide chain, and I'm going to attach it on top of this amino acid that was just added to the A site of the large subunit of the ribosome. I know that this is the right amino acid because the anticodon region of my tRNA matches the codon in my mRNA. And now this tRNA is no longer holding an amino acid. It will be shifted into the E site, and from the E site, it will be ejected. That leaves the A site empty for another amino acid carrying tRNA to enter the A site. This will continue in the elongation phase until you've reached a stop codon. When you reach a stop codon like AUG, you will no longer bind to tRNA, Instead, a stop codon will bind to something called a release factor. And when the ribosome binds to a release factor in the A site, it disassociates the two halves of your ribosome and it just pops off. And now you have this long polypeptide chain that will start to fold up into its secondary and tertiary structure. And then when it's complete, it will be functional and it will do whatever its job is. Fabulous. I've got 3 animations. I feel like it does a good job showing the ribosome scooting down and the tRNA entering the A and exiting the E sites. So check those out. And then I have this overview for us to walk through again. Go ahead and pause the video, and I want you to verbalize what's happening at each step along the way, and then I will do it with you. Okay. We have 2 steps in making a protein. We have transcription, which is the creation of mRNA from the information in DNA. For transcription to occur, we need a promoter, often referred to as the TATA box. Transcription factors bind to the TATA box, and then RNA polymerase II will bind to transcription factors. RNA polymerase II will separate the 2 strands of DNA, and it will make a complementary base pair RNA molecule called mRNA that will match the template strand. So we're complementary base pairing to the template strand, not the coding strand. From the template strand, you will continue, this is the elongation phase, until you hit the termination site. In the termination site, RNA polymerase II will disassociate with the DNA, and you now have an immature mRNA. That's what's being shown right here by this red ribbon. Our immature mRNA has introns, which we have to get rid of. And we also have to add a poly A tail and a 5' cap. When we do both of those things, we've spliced out the introns. We are then left with a mature mRNA that can leave the nucleus. So it'll leave the nucleus via a nuclear pore, and it will go to a ribosome. The ribosome will sit on the mRNA, and the center of the P site will be situated over your start codon, AUG, which always codes for methionine. So each protein is going to start with MET. Once you have MET right in the center of your large subunit at the P site, you can add new tRNAs to the A site, making sure that the anticodon region matches the codon in the mRNA. Each tRNA will be holding an amino acid that's charged and ready to be added to your polypeptide. The ribosome will shift down. Whatever was in the A will move into the P site. The polypeptide will reattach to the top of the amino acid in the P site. And whatever was in the P site is now empty, and it will be ejected from the E site. You continue this way until you have your entire polypeptide chain formed. Then you hit the stop codons on your mRNA. Remember that you have 3 stop codons, UAA, UGA, and UAG, which I remember because you are awful, you are gross, and you go away. So 3 ways to remember stop. One of those 3 stop codons will be at the end of your mRNA. When it is in the A site, instead of binding to a tRNA, it will bind to a release factor, which will disassociate your ribosome and will allow the polypeptide to break away and fold into a functional protein. Here are our beautiful codon charts that we use to figure out what codon codes for what amino acid. There is a trick to using both of these, and you can figure out which one you like best by practicing with them. If we want to use the square one, we would first identify what the first letter of the codon is. Let's say that it's a U. That means that I'm going to be in this row here. Then you identify the second letter. Let's say it's a C. That tells you that you're going to be in this box here. And let's say the last letter is a C. You look at this, the third letter. It follows the same pattern, UCAG, UCAG, UCAG. So it'll be UCAG, UCC as a codon here, codes for serine. So in the square one, you follow each of these sides. First gives you the row, second gives you the column, and third gives you which bar inside of it. Let's pretend that you have the codon AAA. Go ahead and find which amino acid that codes for. First letter, A. So I know I'm going to be in this row. Second letter, A. I know I'm going to be in this box. And then my last one, A. I am coding for the amino acid lysine. Here is the circular amino acid chart. To use the circular amino acid chart, you go from the inside out. So let's use AAA again. You find your first letter here. So I'm going to be in this quadrant. Here's my first letter. Then you find your second letter. I'm going to be in this ray. And then you find your last letter here, A, it's lysine. So AAA, lysine. Let's say that your next one is UAG. Go ahead and find that on the radial amino acid chart. So UAG, that's our stop codon right here. So from the inside out on the round one and from the sides down on the square one. All right, here's practice for you. We have a DNA sequence up top. This is the template strand, so we are going to use this strand to create our mRNA sequence. Take a moment, translate it into mRNA, and then transcribe it into amino acids using that amino acid chart. Here's our transcription. What binds to T? A. And remember, this is mRNA, so what binds to A? U. What binds to C? G. Keep going. And then we end with stop codon UAA. Now let's translate this into amino acids. First one is methionine. It's going to be a start codon. AUG. There's met. The next one is GGG. So GGG. That's going to be glycine. Then we have CAU. CAU. That's cysteine. All right, so those are the amino acids that are sequenced by this mRNA. I'm going to go ahead and delete those, and then I'm going to give you another practice opportunity. You are going to watch this video, so it's linked to there. It's about rock pocket mice. They're really adorable. It's a great video, and it's going to introduce the mutation in the gene that controls their color. You're going to have a practice worksheet, and you're going to use this practice worksheet to do transcription and translation. So here I have the gene for a dark colored mouse and a light colored mouse. Based on the video you watched, you'll see the DNA sequence here. This is the 15th amino acid. That means that this would be the 16th, this would be the 17th, this would be the 18th, and you'd continue on until you've done all of them. That's an 8. You'll look at the DNA and then you'll figure out the mRNA pair. So what's the mRNA pair for this? It will be A, C. So just using my complementary base pairs. This is mRNA, so it will be U, C, C. And then you will use your mRNA to figure out the amino acid sequence. For AAA, we know we're going to be in this first column, this first row, and we're going to be in this box because our second letter is A. AAC right here, coding for ASN. All right. You'll do the same thing for the next amino acid, UCC. So U, column C, find UCC. That's for serine. And then you'll continue until you figured out the entire amino acid sequence for these two different genes. After you figure out the amino acid sequence, the worksheet is going to have you identify what the mutations are, where they are, and how they are changing the phenotype of these different mice. I hope that was helpful. Email me anytime with questions. And Parker and Max say to have a good day. Bye.